So, I welcome you back to another important lecture. And for today, we will see that how come the phloem vessels that we have in case of the plants, they would be moving the sucrose and the amino acids, or they would be translocating the sucrose and the amino acid. But before we proceed with the functionality of these phloem vessels in context of the translocation, our idea of the source and the sink, it should be very much clear. Because wherever the flame vessels would be coming, we would be saying again and again that whenever there would be the movement or the translocation of the sucrose and the amino acid from the source to the sink, so this is translocation. So before going with that mechanism of translocation, I will clear your concept of source and the sink that we have here. Now, I would make it very easier for you. You just have to be understanding is that the source refers to that part of the plant from where we would take the sucrose and the amino acid away. So that would be the source. And the sink would be the that part of the plant to where we would be moving the sucrose and the amino acid. Okay, and alternatively, at one point, the source can become the sink and the sink can become the source. Like if I were to give you a reference, so you see that initially the plants that we have got they either grow from the root tubers or like root tubers mean that if we have got a potato, so this is a root tuber, so the plant they can grow from the root tuber, or these plants they we know that they also grow from the seeds that we have with us. Now you would agree with me that initially the young plant which is growing out of the root tuber in case of the potato, so when this is initially growing, so it would not be photosynthesizing there and then until or unless it would be reaching its appropriate size. So for initially the root tuber that I have with me, that is basically the potato tuber. So this would be acting as a source because from that I am moving the sucrose and the amino acid to the newly forming leaves that I have with me. Likewise, if I were to code you the reference of the seed, so initially the seed that we have with us, so this would be acting as a source because when the young plant would be growing out of it, so it would not immediately be making its sucrose and the amino acid by the photosynthesis, but rather than it would be taking it from the seed that we have with us. Later, when this plant would mature, it would form the fruit and it would again start making the seed. So in that way, the leaves they would now become the source and the seed would become the sink. Okay, now if I were to tell you a situation like there is a fully grown plant and that fully grown plant have the leaves in them and that would be the growth season for them. So they would make a lot of glucose and fructose and by that they would be making the sucrose out of it. So they would be acting as a source because from these leaves we would be moving the sucrose and the amino acid to the root tubers or the stem tubers and over there we would deposit them. So at that time these root tubers or the stem tuber they would act as a sink. And I would say that okay, fine, the growth season has ended and the top of the plant has died. Now, when the top of the plant would die and the next growth season would come, so this root tuber or the stem tuber we have got buried under the soil, and in that season, that this would now be acting as a source, and then plant growing out of it that would be acting as a sink. And then the things could also reverse during the daytime and during the nighttime. During the daytime, the leaves they would be acting as a source because from them we would be moving the sucrose and the amino acid to the storage organ of the plant. And when the night comes and the leaves they stop photosynthesizing, so we have to be breaking these sucrose in the form of the starch in which they would be stored in the sink. And then when we move them back toward the leaves, so at that point at the night, the underground you could say organs that we have got in case of the plant like the root can act as an underground storage organ, so in the night that would become as a sink and the leaf that we have got this would become as a source. So the idea simply is that the part of the plant from where we would move the sucrose and the amino acid away, so that would act as a source, and the point to which we would be depositing the sucrose or we would be storing the sucrose in the form of the starch, so that part of the plant would become as a sink. So here we would see that how come this mechanism of the translocation would be brought about by. So now you see that word translocation is that this is the movement of the assimilates, and by the assimilates what we mean is the sucrose and the amino acid, and when we try to move them from the source to the sink, so this is referred to as the translocation thing. Now the situation is that in the phloem vessels that we have, so these are the phloem sieve tube elements. And in these phloem sieve tube elements, I have to be bringing the sucrose and the amino acid. And these phloem C tube element, they are closely connected with these cells, and these cells, they are the companion cells, and these companion cells further, they are lining with the palisade and the spongy mesophyll cells that we know are photosynthesizing and making the sucrose and the amino acid. So for the spongy and the mesophyll cells, they would capture the light, they would make the glucose, and afterward they would make the sucrose out of it. Now I have to move this sucrose from the photosynthesizing cell to these phloem sieve tube elements. So what happens actually is that these companion cells, since they have the abundant mitochondria in them, and these abundant mitochondria, they would release ATP, energy out of it in the form of ATP. And by that, we would move the sucrose and the amino acid into these companion cells. And once the sucrose and the amino acid, they come in the companion cells, so their concentration in the companion cell would increase, and their concentration in the phloem sieve tube element comparatively is less. So the sucrose and the amino acid, now they would come from the companion cell into the amino acid. Now you think of the situation that in the phloem sieve tube element, when we try to be depositing the sucrose and the amino acid, so it would lower the water potential in them. And when the water potential would be lowered in them, then surrounding the phloem vessels or the phloem sieve tube elements, we have the xylem vessels. And from the xylem vessels, the water would move here into the phloem sieve tube element. And at this point, this movement of the water by osmosis, this would create a very high hydrostatic pressure. And when here high hydrostatic pressure is created, then that would be a driving force that would move or that would push the sucrose and the amino acid toward the sink of the plant. And when I have brought the sucrose to the sink of the plant where I have to deposit them, so I would unload the sucrose. And when the sucrose would be unloaded at the point of the sink, so it would be deposited in the form of the starch. So that part of the plant from where we are taking the sucrose and the amino acid, that is the source. So we are doing the loading of the sucrose and the amino acid. And that part of the plant to where we are taking the sucrose and the amino acid to what we are referring as the sinks over there, the unloading of the sucrose would happen and the sucrose would be stored in the form of the starch. So if I were to tell you the points that you need to remember in case of the mechanism of the translocation, so what we have to be understanding is like these palisade cells, they would photosynthesize and they would make the glucose. The glucose would combine with the fructose and we would form sucrose out of it. The companion cells will then release energy by the respiration, and that would move the sucrose and the amino acid from the palisade cells to the companion cells. And now from the companion cells, when the sucrose and the amino acid, they would reach the phloem sieve tube element, so they would lower the water potential in them. And then the water would move from the surrounding xylem vessels, and then it would generate a high hydrostatic pressure. And under the influence of that high hydrostatic pressure, the sucrose and the amino acid would now be moved toward the sink. At the sink, they would be unloaded and they would be stored in the form of the starch. So I hope you would be clear with the mechanism of the translocation. For the next class, we will see that how come the water is moved from the root toward the leaves. So see you in the next class.